You are working on cell competition. What is this phenomenon and why is it important?
So cell competition is essentially a Darwinian way of thinking about tissues where they are comprised of cells that are not all equal to one another. Some cells are more fit than others. The more fit, so-called winner cells, compete and kill off the less fit loser cells, so that at the end of the day, the tissue is comprised of only the most fit cells. And so why this is important, I think, is illustrated by this really cool example from fruit fly wing development, where we find if we block cell competition, you end up with flies that have unevenly sized wings, which means that these flies are a little bit less good at flying than their sort of wild-type counterparts, which has serious consequences for their fitness and ability to survive in the wild.
So I did a little bit of research on the history of your field, and mechanisms of cell competitions were discovered in the 70s of the 20th century, and then scientists seem to have lost interest in it until the early 2000s. And I was wondering, why is that? Why did the research? Did the questions asked change, or did the technologies change?
I think a little bit of both. So on the one hand, the technology progressed quite a bit to enable mosaic genetic analyses of tissues, which first came in the fly and then quite some years later in the mammal. And this is really essential to be able to study cell competition within a tissue. But in the last 5 to 10 years, I think what has really driven interest in the field forward is the realization that almost all of our tissues are heterogeneous mosaics comprised of clones of varying fitness that compete with each other. And this effective mosaicism has consequences for our propensity to develop various diseases and function over time. And I think we now can use cell competition as a lens to try to understand how these diverse clonal populations within a tissue interact with each other.
So cell fitness and cell competition obviously have many physiological roles. What do you hope the impact of your work is, or what is the medical potential?
Yeah, so I think if you look to patients suffering from a wide range of diseases, from cancer to many different rare genetic diseases, you can look at the interface between healthy and diseased tissues. And you can imagine that you might have cell competition happening at those interfaces. And the idea is that if you can understand how cell competition works, then you can develop therapies to promote the growth of the healthy tissue at the expense of the diseased tissue. And in this way, I think there's a great potential to use cell competition knowledge to develop potential treatment strategies for a lot of different diseases.
From what I know, there are also possible applications in transplantation medicine.
Yes, yeah. So there's been some really creative and cool studies looking at this exactly in the context of rejection and whether we can sort of harness cell competition to make engraftment more successful.
And why did you decide to become a researcher?
So I think I realized quite late on that I wanted to be a scientist. I wasn't one of these children that was running around in swamps collecting butterflies, like many of my colleagues probably were. I always loved telling stories, though. And I originally wanted to study medicine, which got me into a lab. And upon joining this genetics lab, I had a very charismatic mentor who made me realize that actually telling stories lies at the heart of sort of generating hypotheses and doing a lot of good creative science. And so I think that realization, together with the love that I developed for watching tissue morphogenesis unfold and being in a community with people that were also really excited about this kind of biology. I think when I realized that I could be in this community, I could work on tissue morphogenesis, and I could tell stories about it for the rest of my life. When I sort of realized this, there's no other career for me, and here we are.
So you are now also recruiting PhD students, researchers, and now that you've seen things from a different perspective, what advice would you give your younger self?
I think I would tell myself, which I need to keep telling myself now is to really cherish the beginning of projects and to try to savor that time when things are just starting, because that's actually, when I look back at everything I've done, that's always the part that's the most fun. And I think that really pushes me to keep going is thinking about these early brainstorming phases. And I think me, like many others, I'm always impatient to sort of get to the results, but I think it's really savoring the beginnings of ideas and projects is something I would tell myself from day one.